Okay, thank you all for being here to this new edition, the 2024, the first edition of the Next Research Line Fair event for this new year. As we are used to, we are going to have 3 different sessions, each of them for the specific research line that we do have inside the NECSTLab. So we're going to start with the first research line. So it's going to be IT for life science. Then we're going to have DSC, and finally, we are going to close with the heterogeneous high-performance computing. We're going to have overall 14 talks, and as depicted in this figure, as you can easily see, we are going to have talks that can belong from more than one research line. So the kind of organization that we depict was just a way to read out all the presentation that we are going to have. So without any further delay, I'd like to introduce our first session chair, Eleonora Darnese. She is a postdoc here in the Politecnico di Milano for more than a year now. And she's going to introduce all the talk that we are going to have in the IT for life science research line.
Okay, so hello everyone. My name is Eleonora Darnese, and today I'm gonna present you the information technology for life science as what we intended NECSTLab. So a few words about myself. I have a biomedical engineering background, and then I've moved for my PhD in information technology, and more specifically in computer science and engineering. As Marco said, I'm a postdoctoral researcher at Politecnico di Milano, and my main focus are computer vision and medical image analysis with a focus on image registration and segmentation. So what we do here at NECSTLab that follows the IT for life science paradigm is working on 3 main areas. One is based on image processing and medical imaging. One is related to genomic, and the last one is related to well-being and wellness. So starting with the first area, we have different work. One of them, for example, is Lucifex, that is lung cancer for the lung cancer identification through the radiomic approach. And what we have here is that we have created an automatic pipeline that starts from routinely acquired medical images like CT and PET. And what it does is integrate the information to identify and automatically segment the tumor. Then we extract the radiomic features that are high order and standard feature that we extract from images. And we use such images to create a sort of signature of the mass that is then used by machine learning models to characterize this mass as primary lung cancer or metastasis, and from primary lung cancer among the different histological subtypes. Also in the imaging field, we have AbdemReg that deals with image registration. So with this project, what we want to do is being able to correctly align different stacks of images, one over the other. Here, the main challenge is that we are dealing with deformable image registration. So we are not working on pure rotation and translation, but we can also modify the parallelism of lines. And this is super interesting also because we have applied our framework to the abdominal area that presents huge variability and deformation. So compared to other state-of-the-art solution, we are dealing with a complex task, and we are also able to achieve interesting results in correctly aligned magnetic resonance images, both intrapatient and interpatient in a monomodal or multimodal way. So moving to the more complex part of genomics, we have 2 sorts of approach. One is based on knowledge graph. So we have different projects that are dealing with different steps of the entire pipeline, starting from the construction of the biomedical knowledge graphs to the representation of this graph, and then feature selection that can be based on task-driven automatic methods. And then we have also the embedding and use all this information to perform a various number of tasks like drug repurposing or dementia identification. Also, in the part of genomics, we have worked on different stage of the genomic pipeline, going from the quality filtering till scaffolding and gene annotation, and we have exploited different type of architectures, namely GPUs and FPGAs. As we can see, we have different tools in this domain that can achieve interesting results, both in terms of energy efficient and speedups against state-of-the-art solution, but also they are they can be integrated into tools already there in the state-of-the-art. Last but not least, we have the third area that is dealing with the broad concept of well-being and wellness. And in this sense, we can have OptoSense, that is a prototype mask that uses an optical system to understand how well we sleep. So what we have is that sleep is critical for all of us, and have a good quality of sleep is interesting. And so sleep the detection of the stage in our sleep are super relevant to ourselves. So I won't take more time on this overview, and I will let all our speakers take the stage and go into detail of their project. Thank you. No? Oops. Okay, so I welcome for this first talk, Isabella. The stage is yours.
Okay, so thank you, Eleonora, for your presentation. I'm Isabella, and today I will present to you the topic of my research. But before that, I want to present myself. I'm a second-year PhD student in information technology here at NECSTLab. But despite this, I have a bioengineering background since I've got my master's degree in bioengineering at both Politecnico at University of Illinois at Chicago. It is for these reasons that my research focuses on computer vision, mainly applied to medical scenario. So to get a little more into detail, my PhD revolves around designing a framework specialized to analyze medical images during end-to-end image-guided biopsy procedures, implementing deep learning method at the algorithmic level to aid the end user with more accurate and faster analysis. So given the complex application scenario, I will start introducing you to the world of biopsy procedures. So they are multi-step, multimodal, and multi-scale medical procedures that starts from the acquisition of images of a disease or healthy organ from a patient of interest. And then it proceeds by aligning them to a common reference frame through the image registration step. At this point, the procedure steps into the microscale, where images of the acquired tissue samples are acquired and then analyzed through several image processing techniques to allow the end user, such as physicians or researcher, to produce a medical report. So it is evident that being able to accurately and precisely analyze those images produced could ease the end user to produce a possible diagnosis. Despite this, the current bottleneck in the clinical practice lies in the time-consuming nature of data acquisition and analysis. In fact, due to the massive amount of images that are produced every day in the medical scenario, the manual annotation and analysis have become unavailable within reasonable timeframes. This aspect worsen even further when considering that those images could be of different dimensionality scales or acquired with different modalities and always at different medical centers. So I want to say about 15 years ago, deep learning came around to solve all these issues. in fact, has been demonstrated to be capable of handling huge amount of heterogeneous data at once. In addition, it has demonstrated to ease a human error-prone task with higher image quality analysis. And furthermore, it has also guaranteed faster analysis thanks to its efficient inference step. So it is for these reasons that I decided during my PhD to exploit the deep learning potentialities while building upon its weaknesses to produce a complete image analysis pipeline for a biopsy procedure. In particular, my pipeline starts at the macro scale with the image registration step, and then goes down into the micro scale through several image processing steps. But the key aspect of this work is to focus on the multimodal and multi-scale integration of the image analysis. In particular, different modalities of images will be integrating at both the macro scale and micro scale, while different dimensionality scale of the images will be integrated at the end of the pipeline to close the pipeline and ultimately allow the end user to ease from a complete analysis. So during my PhD, I decided to follow a top-down approach, starting from the macro scale with image registration step. As explained by Eleonora, in this work, we try to get rid of the rigid pre-registration step that usually follow the deformable image registration procedure. In particular, during this step, we design a spatial regularization strategy to fix the translational and rotational deformation components that usually are residual from a rigid or affine pre-registration step. We designed this strategy as a loss function that could also be used with a different neural network architecture and allow our network to learn different ranges of image deformation. We tested our strategy on a unit architecture, and we demonstrate that we are able to reach up to 12% of mean structural similarity index measure over the baselines while getting rid of the image per alignment step. At this point, we decided to step down at the micro scale, and we try to tackle the problem that afflict the histopathology images that could lie in different color domains when acquired with different microscopy scanner. In particular, we design a cycle gun-based architecture to learn a bidirectional mapping and try to bring all the images to lie in a common color domain to then ease the subsequent processing task. Our strategy allows to tackle the problem of having unpaired histopathology images during training, as well as being compatible with the RGB image format. So at this point, one could imagine how our end user would be interested in understanding if the image that is seeing is coming from a diseased or healthy tissue. For this reason, I decided also to deploy some classification and segmentation networks. And in this case, as I focus my attention on synchrotron radiation microCT images, which show a very high intra-class and very low inter-class variance that leads even a human with naked eyes to struggle in identifying differences among these images. For this reason, I started building a semi-supervised strategy to segment microstructures that are then employed to with the image classification neural network to let it focus the attention on the most discriminative features inside our images to classify among healthy and osteoporotic images. Our strategy allows us to reach several improvements, both during segmentation and image classification with respect to the state of the art. but also is capable of being aware in detecting fine-grained microstructure differences among fine-grained images. So building upon this work, I decided to dive even more into the classification step. And I also tried to solve the hidden stratification issue that leads microscale image that usually have a label that is assigned at the macroscale to be instead characterized by different subclasses at the macroscale. For this reason, I built a convolutional autoencoder k-mean-based clustering approach that is able to derive macro to micro scale information matching. And by manipulating the embedded space, I derived the data-driven weights for the classification loss function, as well as data-driven majority voting threshold for the majority voting whole slide image classification. Our strategy allowed to improve the performance reached from the previous work, as well as reaching up to 28% of per class average accuracy from the standard threshold, the image classification counterpart. So during this last year, I've also moved the first step into the multimodal integration, focusing my attention on the microscale dimensionality. In particular, I tried to exploit the intra-domain semantic knowledge that come from training a large histopathology dataset to perform microstructure segmentation in low data regime. In particular, with this strategy, we employ a multimodal transfer learning methodology to predict synchrotron radiation micro-CT mask lacunae in low data regime, and we were able to reach up to 63 dice similarity coefficient osteocyte and lacunae segmentation, while improving the state of the art with 44 times smaller datasets. For what concerns the next step, I'm planning to both improve the multimodal integration at the macro scale with a multimodal image registration and at the micro scale for the segmentation part, while as the last step to integrate also different scale images. In particular, I wanted to study the interaction between different scales in real world data settings, which means with unpaired data, as well as heterogeneous in modality, or when some modality are missing, to ultimately provide to the end user with a complete image analysis pipeline. So I thank you for your attention, and I'm more than happy to answer to your questions.
Thank you, Isabella. I have a few questions, and the first is regarding the hidden stratification issue. You mentioned that you have exploited information from the embedded space to derive loss function weights and majority voting threshold for pathwise and whole slide image classification. Can you briefly describe the strategy you have used to derive them?
Okay, yes. So maybe I can go. Okay, here. So starting from the weights for our cross-entropy loss, I try to exploit those correct matches between the subclasses and the superclasses during clustering. In particular, we exploited the two algorithms for clustering, which are the k-means clustering and the Gaussian mixture model clustering algorithm. So exploiting the proportionality between the correct matches with respect to the all prediction from the unsupervised clustering, I try to weight per class the prediction made by our cross-entropy loss. While concerning the majority voting threshold, I've not just exploited those correct prediction coming from the clustering step. but I also exploited the prediction coming from the Petroid's image classification. So I made some proportionality between the correct prediction of the Petroid's image classification, as well as the ones that in the same time were correct during the super and the subclass matching.
Thank you. I have just another short question. You talk about multimodal integration. So you said you are improving it at the macro scale and the macro scale levels. Can you give some insight on the approaches that you are targeting?
Okay, yes. For the macro scale, at the macro scale, I'm trying to exploit some loss functions that are able to also tackle the issue of a different bit intensity scales between images, in particular exploiting multimodal loss function during the image registration neural network training. While at the micro scale, I'm trying to improve the image segmentation step by employing knowledge distillation strategy. For example, employing prototype knowledge and noise reduction for the feature during teacher and student training.
Okay, thank you so much.
Thank you.
Okay, so we can move to the next presentation, and I'm happy to introduce Guido Walter Di Donato that is presenting Genogra.
All right, so hello everyone. I am Guido Walter Di Donato. I am a postdoctoral research here at NECSTLab, and I am the CEO of Genogra, the startup that is born at the NECSTLab and that is building the first pangenomic analysis pipeline. So today we are here because we all know the importance of genome analysis and the potential impact of this technology in different areas of science, especially in that of personalized medicine, where genome analysis enable early diagnosis and targeted therapy in different medical fields like oncology, cardiology, and rare diseases. The problem is that despite these very good and promising results, today only almost 7% of the patients can actually benefit of genome analysis. And this is mainly due to the fact that genome analysis does not scale very well. From our customer interviews, it emerged that it can take from 10 days to up to 18 months for geneticists to get back the results of genome analysis. And this, of course, is not compatible with the medical practice, but also other application of genomics. And if we go deeper in this genomic workflow, we can easily understand how the secondary analysis, that is the software-based analysis, so the one that is made through the computers, basically, is the main bottleneck of the whole process. This is due to the fact that the available software tools for genome analysis are redundant and error-prone and are very hard to use without offering any ease of usage to the users. This also comes with insufficient scalability of the analysis, also due to the lack of qualified personnel that can operate these tools that are too hard to use. So for all these reasons, today, we are not leveraging the full potential of genomics yet. And to understand why we are not able to do that today, we need to focus on the paradigm of the analysis that is implemented in currently available software. So current solutions are limited in scalability because they all involve comparing the DNA data of our sample with a reference genome that is the same for the whole the species, for example, for the human species. And this comes with a lot of limitations, both in terms of accuracy of the analysis and also, of course, of scalability, given that we all share more than 99% of our DNA. So we need to change the paradigm of the analysis. And starting from 2016, 17, the research community emerged with a proposition. So to move from using reference sequences to reference genome graphs, so entering the era of pangenomics. So instead of using one reference sequence, we can use some pangenomes as reference, so some collections of genomes that contains genomes from various individuals of different kinds, with different features, so from different populations, for example. And this is able to reduce the bias of the analysis and also to reduce the redundancy of the analysis, because we can focus more on the variations that are the actually informative part. And in particular, we are able with genome graphs to represent genomic variability in the closest way to the actual biological reality. So we are able to represent in a very precise way some structural variations, some copy number variations, and so on, that are very difficult to target with string-based softwares. So just to wrap up, the advantage of this new data representation is the great scalability, the optimal quality of the analysis, and the unexplored application that this kind of representation can enable. For example, studying how genomes evolve during time. So this is the reason why in Genogra we are currently developing the first pangenomic analysis platform. It's a software platform that is usable both for research and clinical purposes, and that is completely based on genome graphs. So it implements this new paradigm of the analysis, and thus it offers some much faster and easier workflow of analysis that involves much less tools, while also being more accurate. And this is all possible thanks to our core technology. Our core technology is basically made of two things. The first part is a way to represent and store the genomic information together with all the metadata that are associated to the sample, included in our reference graph. And the second part, that is the actual core of our technology, is a sort of search engine, a sequence to graph aligner that is able to map DNA sequences of interest onto the genome graphs in a very efficient way, leveraging hardware accelerators. In particular, we are using GPUs to speed up the process in a completely transparent way. And this is patent pending, both in Italy and at PCT level. So this technology allows us to be very competitive with respect to our competitors in the market on a lot of different factors that emerged as value drivers from our customer interviews. So for example, the possibility to do population genomics in an efficient way, or having both high performance and the highest quality of analysis provided by among all the competitors. So this places us in a very competitive position in a market that is growing really fastly. The genome analysis market size, this relates just to software and services for genome analysis, is growing really fast. Is actually one of the fastest growing market overall globally. And it is, and forecasts are that this market will reach in 2028 almost 12 billion of size worldwide. We are focusing on the European market that is almost 25% of the total market, and today is almost 1 billion, and it is expected to grow up to almost 3 billion in 2028. But the most important thing is not how the market is growing. Of course, this is a positive thing, but the actual informative part is that the market is segmented in three main customers. So we have diagnostic applications, medical applications. We have base research. So we target, in that case, customers like university or public research centers. And then we have applied research, especially in the field of biotech and pharma. And our technology is actually so flexible that we can target all these markets, all these segments, but we also developed a go-to-market strategy to validate as soon as possible the technology on the market while reaching the most regulated market. So our plan is to start with the non-human research. So we don't need any compliance, GDPR compliance, and so on. We just need to protect the data of our users. And this is quite simple. There is off-the-shelf solution to do that. So we just need to integrate that. Then, after getting the GDPR compliance and guaranteeing a safe processing during all the stage of the processing of the genomic data, then we can move to human research. And finally, after getting the in vitro medical device regulation, we can get to the diagnostic market. Of course, to reach these diverse segments of customers, we also need a flexible business model that can adapt both to the user's need, but also to the customer's need, because in practice, these two figures are not the same. So as user needs, we need to understand which are the tools, the services, and the kind of interface and experience that is best suited for our kind of users. On the other hand, we need to satisfy the customer's needs. So the number of users that need to access the platform, but also the kind of deployment. So you want the deployment on cloud or on premise. We support both, but of course, it changes the subscription fee and our business model. So moreover, an important thing is that Genogra is using an open core model. So basically, we not only develop some proprietary tools and algorithms that we integrate in our premium platform, but we also contribute to existing open source projects that then we integrate in our platform. We also develop utilities that we make available to the community. This is to provide also in a free way, in an open way, some contribution to the fields. And so to conclude, let me present the team behind Genogra. I'm not alone in this adventure, and I'm lucky to have a great team with Alberto, Guglielmo, Mirko, the Professor Sant'Ambrogio, of course, and also Alessandro in the part of business development. And we also have two advisors that are helping us on a strategic point of view and on medical applications of our technology. We are a very technical team. Our competencies are very tightly related to the high-performance computing, the genome analysis, and the graph analysis world, and we developed our expertise working not only at Politecnico di Milano and at NECSTLab, but also in collaboration with other institutions, university, and also companies, both in Europe and in the United States. To conclude the presentation, let me show the current status of Genogra. We are right now, after 3.5 years of research and development, we are at TRL 5. We have an MVP that is available. So the prototype of the platform is available and has been already validated by 2 geneticists. Right now, we are looking for seed funding to reach TRL 8. So we are looking for 600K. But of course, I'm not here to look for investors. Of course, if you want to invest, just drop me an email. But the reason why I'm here is that to reach TRL 8, we need to validate our technology. So if you process genomic data, if you are working on some application involving genomics and genomic data analysis, then maybe we can be a partner to do some pilot study where we can use our technology, our tools, to look if we can achieve some better results with respect to currently employed practices. And so we can both benefit from this pilot study. You can find maybe a more promising solution for your problem, and we can find a promising application of our technology. So with this, I'm concluding my talk. Thank you for your attention. Here you can find our website, genogra.com, and our email contacts. So just drop us an email if you want to collaborate or we are interested in investing in us. Thank you.
Thank you for sharing Genogra with us. I have a question, and it's related on the difficulties that you have faced to bring research outcome to the market.
Oh, this is a very interesting question, and probably is one that I cannot answer in a complete way because we are not on the market yet. So we are still in the process of this technology transfer process. So we are trying to bring the results of our research on the market. And I think the most important thing is for a researcher to be an entrepreneur is a sort of change of mindset. So as a researchers, we are often involved in some activities that allow us to bring new knowledge to the world, to advance the state of the art of just a tiny bit, but it's very important. It's a very important contribution. And we always tend to, let's say, sell our work just after achieving the results. We do the work before, and then we try, for example, to valorize it with a publication and so on. What you need to do when you move to the entrepreneurial world, let's say, is probably start selling before developing. And this is a complete change of mindset that is not easy for us researchers, because we are not feeling okay if we have not developed the technology that we are trying to sell. But actually, in a startup creation process, probably you need to find the product that can be sold and then starting developing it. So is this change of paradigm, I think, that is the main difficulties and the main differences between doing research and doing startups.
Thank you so much, Guido, for the presentation and for your answer.
Thank you.
Okay, so I think that we can move on on the third presentation of this evening. And I will call to the stage Mirko, that is presenting his work.
Okay, thank you, Eleonora. Hi, everyone. My name is Mirko, and today my talk is about pangenomics, and in particular on how a graph-based approach will improve genome analysis in the next few years. So before getting deeper into this talk, I want to leave you some notes about me. So,
Sorry, just the screen sharing has gone off. And that's...
okay, carrying on. So, as said, my name is Mirko Koch, and today I'm talking about pangenomics, and in particular on how a graph-based approach will improve genome analysis in the next few years. So before starting, I want to leave you some notes about me. So my journey in this field of genomics started during my bachelor and master degree in biomedical engineering, where I discovered to have a great interest in computational genomics, and also in creating optimized solution which leverage the hardware acceleration, and also, and in particular, in pangenomics, which is the study of multiple genomes at the same time, like analyzing the genome of a population, for example. And this drew me here today because currently I'm a PhD student here at Politecnico di Milano. And as my colleague Guido said before, I contributed to the foundation of the startup Genogra, whose vision matches my research interest, which I will explain in this talk. So, talking about computational genomics, everybody knows that this field has gained a great importance in different contexts, ranging from the personalized medicine to the radiomics, to the metabolomics, the live and feedstock industries, and many, many other contexts. But despite this great amount of application, we can see that the genomic pipeline is pretty much standardized, because it always starts with the taking of the DNA sample, which is then sequenced to obtain subsequences of the original genome, and then the computational task begins, because this reads needs to be assembled together to recreate the original genome, which is finally analyzed to find some specific sequences of nucleotides inside it. So until 2015, both the assembly and analysis task could only be supported by a linear representation of the genomes. And this led to the creation of a consensus reference sequence, which was made by merging a multitude of genomes into one single sequence by selecting for each genome site only the most frequent variation among all the possible variation for that site. And this approach introduces a great limitation, which is called reference allele bias, because the data incongruent with the referent allele is underreported. And in particular, during the read mapping process in resequences, this results in a systematic misrepresentation of the authentic sequences that build the reference. And moreover, this approach can also limit the scalability when we have to deal with a population of genomes. And in the end, this is also inefficient to represent the structural variation, so the bigger variation inside the genome. For this reason, in recent years, genome graphs has been proposed as a more efficient and a more compact way of representing the inter and intra-individual variability because of their ability to encode variations as alternative paths inside a directed graph. The problem here is that graphs are more complex structures than sequences, and this introduces new technical challenges when we have to develop graph-based computational pipelines for the genome analysis. In particular, the main challenge regards the research of specific sequences or patterns inside a reference, which has usually been called the read mapping process, but that in case of graphs is the sequence to graph alignment process, which consists of computing the alignment of the query sequence to a path in the graph, having the maximum alignment score among all the possible paths. Well, the successful execution of this task is biased by different factors. For example, the accuracy and completeness of the results, the flexibility of the tools, and also the performance improvements on top of the alignment algorithms. So in order to investigate the trends in developing this type of tools, so sequence to graph aligners, I implemented a novel methodology for a comprehensive analysis of the state-of-the-art tools. In particular, this methodology allows for a qualitative analysis of the three factors I mentioned before, where each factor has been decomposed in a set of parameters that collected together allow for evaluating the aligners on specific data and in specific case studies. On the other side, I also implemented the first open source benchmark suite for this type of tools to quantitatively analyze the accuracy and the execution time. In particular, the benchmark suite is completely automated to test the performance of the different aligners over a pool of data, which comprises both the reference graphs and the reads to be mapped to these references. And I think it's okay. So in my first version of this work, I included 7 state-of-the-art tools, which every one of them promises to solve any critical issue. But however, I noticed 3 crucial aspects in this study, because I discovered that there isn't a solution with outstanding performance on all type of data. But different tools perform better on data with different characteristics. For example, one of the main characteristics is the linearity of the reference graph. For example, some tools go faster on more linear graphs and other tools on highly branched one. Then another fact is that these tools are slow. So there is no solution integrating hardware acceleration solution, for example. And in the end, a crucial aspect is that next generation sequencing technology allow for a faster genome sequences, which increase the production of genome data. And this allow us to create larger and larger graphs, but current solution can't handle such big data. So these highlights helping me in defining the scope of my PhD thesis, It is the creation of a definitive framework for graph-based genome analysis to solve the issue of current solutions. And in particular, I thought about a framework which is composed by 5 tasks, which starts with the graph partitioning to break the original large graphs into smaller subsections according to the branching density. Then there is the selection of the best aligner for each partition based on the characteristic, of course, of the partition. And after that, the main task is the execution of the alignment, which will be done by using a already optimized solution present in the state of the art, or with new promising algorithms that I will optimize by myself. After that, there will be a need to manage the cross-partition alignments, and in the end, the merging of the results of this hybrid alignment. In particular, the first, the third, and the fifth task can also benefit from hardware acceleration with parallel computing on GPUs with the obvious aim of speeding up the process. So currently, I'm mainly focusing on the third task, so the alignment execution, because it is the more computational intensive, and also because different analysis pipeline needs different features and different fine-tuning of all the possible parameters. For this reason, I recently developed a new implementation for the GWFA algorithm, so the graph wavefront aligner algorithm, which is a really promising strategy for the sequence to graph alignment because of its outstanding performances in terms of execution time. So, however, the first implementation of this algorithm lacked the computation of the alignment tracebacks, so the precise sequence of edits needed to map the reads on the graphs. For the reason, I re-implemented this algorithm, allowing a more complete retrieval of all the alignment results, and so making it usable in real case studies. On the other side, I'm currently working on a GPU implementation of the GEDMAP aligner. GEDMAP is another interesting tool that tries to solve the problem of memory occupancy of graph structure, because as I already said, graph structure are more complex, so they need more memory to be stored inside the machines. And in particular, this tool converts the graphs into elastic degenerate strings, obviously reducing the size of the data structures, but increasing the execution time. And that's why I'm focusing on implementing another accelerated solution using GPUs to, of course, speeding up this process. So, well, creating tools, of course, can have a great impact in genome analysis, but I think it's not enough, and that's why I'm here. So I proved that the efficacy of my solution on public data available from open source databanks, but I aim to do a little bit more, because my idea is to use graph-based genome analysis pipeline in real case studies and scenarios, and that's why I'm looking for collaboration with bioinformaticians to identify new possible application of genome graphs in the most used pipelines, and also to create end-to-end solution that could really enhance the genomic analysis, unleashing its full potential. So with this, I have concluded. Thank you for your attention. And if you are interested in a follow-up, don't hesitate to reach me out.
Thank you, Mirko. So I will go with a quick question. And you said that sequence to graph alignment is biased by accuracy, flexibility, and performance. So how did you decompose these factors to evaluate the state-of-the-art tools?
Okay, thank you. So for what concerns the accuracy and completeness of the results, the parameters that needs to be analyzed to assure that the accuracy is enough is, of course, the supporting of cycles inside the reference graphs, but also the, as I already said, the computation of the traceback, because without this feature, we are not able to use the tools in real case studies or real case scenarios. On the other side, there is also a point on the completeness of the results, and in particular on the standardization of the input and output files. And so the research is pushing the idea of adopting the GAF format for the output of the alignment and the GFA format for the input of the alignment, because these type of formats are the more complete present in the state of the art. Then, for what concerns the flexibility, two factors that are fundamental inside a tool are the possibility to have a custom scoring matrix to adapt the different tools to different scenarios, and also the possibility to have an FI gap penalty. On the last side, the performance improvements, the most interesting solution integrated an inter-sequence parallelism, usually leveraging multi-threading on CPUs, but there is an interesting push to adopt GPU's implementation for this task, so to spread the computation of different sequences on parallel devices. Then another fundamental aspect is the use of heuristics. In alignment heuristics, like banded alignment, which reduce the compute space, and also seed and extend heuristics, which reduce the execution time by computing the alignment only of a subsequence of the reads we want to find, and then to extend this subsequence in a second time.
Thank you so much, Mirko, for your answer and the talk.
Thank you.
Okay, so we can now move to the talk presented by Susanna Bardini, and I leave you the stage.
So good morning, my name is Susanna Bardini, and today I'm going to present you OptoSense, a prototype of mask for sleep quality monitoring. So first of all, just a brief introduction. I'm graduating in biomedical engineering, and my research area focuses on wearable devices for personal well-being monitoring, and I'm willing to start a PhD within this area. So we can say that nowadays we are quite used to collect several physiological parameters in order to increase our personal awareness for what concerns our habits related to physical activity, nutrition, stress management, and sleep quality. And in particular, today we will focus our attention on this last one, so on sleep monitoring, as it is of fundamental importance in our everyday life, as it is crucial for several cognitive and metabolic functions. Traditionally, sleep monitoring is tracked by identifying the sleep stages that characterize our rest, and in particular, light sleep, REM, and deep sleep stages that together with the awake phase are usually represented in a graph like this, in which we can appreciate the time percentages that we spent in each sleep phase. But let's now go deeper into the techniques that allows us to identify the sleep stages. We have several tests that allows us to identify and to monitor the clinical sleep disorders that might affect. And even if we will not focus today on this area, we can mention polysomnography and actigraphy as the two tests that today represents the state of the art. We have then also several devices that allows us an autonomous at-home monitoring and that provides us with more immediate feedbacks. We have devices that do not even require the direct contact with our body, but they are meant to be placed near our bed or under our mattress in order to track our snoring or our body movements during rest. And then we have also wearable devices that aims to continuously collect biosignals related to brain or muscular activity, heart rate, or eye movements. And examples of such devices are, for example, headbands or smartwatches, rings, or even sleep masks. And in particular, sleep masks are gaining an increasing attention in recent years' research, as they are particularly interesting as on one end allows to limit the possible external noise sources that might affect our physiological sleep, so it's, for example, the ambient light, and on the other hand allows to embed systems that are less invasive and that aims to monitor heart rate or eye movements. And in particular, eye movements is a technique that is gaining an increasing attention because it seems to be particularly feasible for this kind of monitoring. And traditionally is exploited with electroculography, that however, it is not exploitable in our everyday life as it is quite invasive and requires previous medical knowledge. And so in order to correctly place several electrodes, and so it is not feasible for a daily monitoring. And for these reasons, there are studies that aims to find more feasible solution for a daily monitoring based on optical systems or on magnetic sensors. And it is within this context that I had the idea to implement a prototype of mask based on an optical system that is less invasive, less bulky, do not require any previous medical knowledge to the user, and it does not affect our physiological sleep. So here we have a picture of OptoSense. As I just mentioned, the prototype is based on an optical system that is composed by an infrared LED and 3 photodiodes put around it. The working principle is basically based on the emission of an infrared light that is reflected on our eyelid and detected by 3 photodiodes. And to date, the device works on the Arduino Nano RP2040 Connect. So OptoSense has two working modalities. The first modality is the calibration one that allows the user to verify at the first usage of the mask that the device is correctly placed on our face, and in particular that the optical system is centered with the center of our eye in order to allow a proper monitoring. So the user can verify this by checking for the match between the real direction of displacement of the eye under the eyelid and the output printed on the screen. And such function has been implemented by associating to the evolution of the signal over time, the possible direction of displacement of our eye under the eyelid. The second modality is the actually monitoring modality that continuously record the mean value detected by the 3 photodiodes that actually corresponds to the real mean displacement of our eye. And in order to associate to such signal the different sleep phases, I've developed a post-processing algorithm that was initially based and parameterized starting from the classification performed by the Samsung Galaxy Watch 5 that I initially exploited as ground truth. But let's now go to analyze the structure of the algorithm. It is organized on two parts. The first part allows to identify the deep sleep, REM, and light sleep phases, while the second part allows to discriminate the awake stage. So starting from the signals collected across different recordings and among two different users, I have identified patterns that were characteristic of the different sleep phases. And in order to properly classify such phases, I first of all normalized the signal. I reduced the noise of such signal, and finally, I've set experimentally two thresholds that I've compared with the variance of the signal obtained so far in order to discriminate the light sleep, REM, and deep sleep stages. At this point, however, the algorithm wasn't able to discriminate the awake stage from the light sleep phase, as we can observe the two patterns differ only for the frequency of the signal. And so at this point, the algorithm starting from the samples belonging to the intervals already classified as light sleep stages, computes the Fourier transform in order to be able to associate the intervals characterized by a peak at higher frequencies to the awake stage and the intervals of the signal characterized by a peak at the lower frequencies to the light sleep phase, in order to be able finally to correctly classify the 4 stages that characterize our rest. So here we can see a summary of the steps that compose the algorithm that I've just described, organized on the two parts. Then at this point, I found interesting to compare the classification proposed starting from the optical system on which actually the working principle of OptoSense is based on, with another classification carried out also within the same device, but starting from the gyroscope data, in order to be able to prove the internal consistency and coherency of the device. For this classification, actually I focused my attention on the identification of the light sleep phase, because to date, the second technique is quite noisy, and so I had to take into consideration the breathing rate and the body movements that in the light sleep phase are more constant in time and limited, and so it was a feasible phase in order to perform such comparison. So here we have an example of light sleep classification starting from the data recorded by the optical system, and an example of light sleep classification starting from the elaborated gyroscope data. We can see that the two classifications are coherent, and in particular, in order to identify the light sleep phase starting from the second classification technique, I have associated the light sleep phase to the portion of the signal that overcomes a threshold that I have experimentally set respect to the rest of the recording where the signal remains under such threshold. So summarizing what I've described so far, I started from the classification performed by a commercial device. Then I developed my own algorithm based on, in order to classify the sleep phases starting from the data collected with the optical system. And finally, I proved the internal consistency and coherency of the device by comparing such classification with a classification performed starting from the gyroscope data, focusing on the identification of the light sleep phase. And for what regards the light sleep time computed, starting from the gyroscope data, we can say that this last classification somehow overestimates sometimes the time spent in the light sleep phase respect to the classification performed starting from the optical system and the ground truth data. But however, it was interesting to have such comparison of two techniques exploited within the same device. To date, OptoSense provides a feedback to the user, so we can observe that we have a qualitative feedback from which we can observe the time percentages that we spend in each sleep phase. And then we have also the possibility to download a file reporting such data. And also we have a comparison with the typical time intervals reported in the state of the art. So why OptoSense? We can say that OptoSense is a device that is less bulky, less invasive. It is easier to use because it does not even require previous medical knowledge to the user, and it does not affect with our physiological sleep. And for what regards possible future works, I'm actually working on the substitution of the Arduino Nano with the NECSTLab Sense Me that is characterized by a smaller form factor and a wider range of industrial grade sensors. And moreover, I'm working on the implementation of real-time classification in order to be able not only to provide a feedback to the user, but also to interact with the user itself. And finally, I believe that it might be interesting also to add the possibility to modify the orientation of the infrared LED and free photodiodes in order to overcome the rigidity typical of the mask structure. So finally, starting from this work, I believe that it might be interesting to expand this project in order to be able to evaluate how the environment affects our sleep quality, and also to be able to evaluate if the sleep phases that occurs during our rest somehow affects the feeling that the user has of his rest. And this on one end. On the other hand, I believe that it might be interesting to evaluate also the interaction between sleep and our cognitive performances and stress management, and also the relationship between our sleep and our physical activity and nutrition habits. So thank you for your kind attention, and I will be more than happy to answer to your questions.
Thank you, Susanna. So you have used like real-time classification. So which are the advantages of use such type of classification?
So yes, thank you for your question. I believe that exploiting a real-time classification could open several advantages and several new possibilities. For example, it might be interesting to be able to connect OptoSense to an alarm clock in order to interact with the user itself in real time. So for example, at the awakening time, if the user is in the REM phase, the alarm clock could be postponed in a predefined time range of, let's say, 10 minutes, in order to allow the user to wake up in a more appropriate sleep phase, for example, in the light sleep phase, in order to provide the user with a better feeling of his quality of sleep.
Okay, thanks. And up to date, the classification is based on the data collected with the gyroscope as being exploited for the validation of the classification of your optical system. So do you think that it could also be exploited to bring added value to your solution?
Yes, so yes, to date it has been exploited only to validate the system. I believe that by exploiting the NECSTLab, it might be interesting to introduce a standalone classification based on the gyroscope, or maybe in combination with an accelerometer or other motion sensing sensors, in order to be able to provide a sleep classification based on two different techniques within the same device.
Thank you so much, Susanna.
Thank you.
Okay, so we have come to the last but not least presentation of this evening, and I will leave the stage to Beatrice. You can share your screen.
Okay. You should be able to see my screen now.
Yep.
Okay. Thank you, Eleonora, for the introduction. Hello, everyone. I am Beatrice Branchini, and today I'm going to present you a GPU approach that aims at improving performance and flexibility of a prototype caller. Firstly, let me introduce myself. I am a second-year PhD student in information technology at Politecnico di Milano. Currently, I am a PhD intern in the HPC group at the Pacific Northwest National Lab. But differently from one can expect, my background is not in computer science, but is in biomedical engineering. And this is the reason why research for my PhD focuses on high-performance computing techniques applied to the genomics domain. So in finding ways that are capable of speeding up the execution of the computation of genomic workloads. And this project is perfectly in line with my research topics. We are in the domain of personalized medicine. Personalized medicine is one of the most cutting-edge approaches in the healthcare delivery field. And the goal of personalized medicine is to provide therapies that are tailored to the patient's disease, to the patient's need, in order to specifically address all the healthcare problems. Among the resources used in personalized medicine, there is the patient's genome that has to be computationally analyzed in order to highlight those peculiarity and find those information that are crucial in order to develop drugs and to formulate specific diagnoses. In this context, variant calling is the process that aims at identifying small variation within genomic data, and for example, single nucleotide polymorphisms, so single base variations, or insertion or deletions in the genome. The state of the art offers a large variety of tools for mutation identification, but the gold standard for variant calling is haplotype caller contained into the genome analysis toolkit developed by the Broad Institute. The pipeline of this tool is composed of 4 steps. The first is the identification of active regions, so those regions showing a higher probability of having mutations. Then the haplotype assembly, so the assembly of those mutations that are more likely to be inherited together. Then, having those haplotypes, the tool aligns the reads against the haplotype with an algorithm that is called PRHMM. And finally, the tools concludes by doing genotyping in order to assess whether a mutation is present or not. If we take a closer look at this pipeline and we analyze the execution time for this tool, we can see that the third step represents a major bottleneck. This step, that is the one based on the PRHMM algorithm, can take up to the 96% of the entire execution time. So only to align the reads against the haplotype, the execution time dedicated to that portion is almost the 96%. So why do we have this bottleneck? Mainly because of the large amount of data that this tool has to analyze at each run. But also, this is combined to CPU inefficiency when dealing with such workloads. General purpose CPUs are not capable of handling such a large amount of data and analyzing that in a short amount of time. And time is a crucial component when we come to healthcare delivery. So the state of the art already offers some solutions that aims at speeding up the execution of the PRHMM. And this is done by offloading the computation to hardware accelerator to meet real-time performance requirements. The problem with all these solutions is that they are fundamentally flawed and we cannot use them in practice. And this is because they lack of flexibility when dealing with input data. They cannot analyze all the sequences that might have to analyze. So this means that they cannot be used in standard practice. For these reasons, in this talk, I will present you a novel high-performance GPU-based accelerator for the PRHMM that features an optimized load balancing methodology that allows our accelerator to process sequences regardless of their length. Furthermore, we developed a methodology that enables a seamless integration within the tool and optimizes the data transfers between the host and the device. In this case, we consider the dynamic programming formulation for the PRHMM, which is the one already used in haplotype caller. And in order to efficiently implement an algorithm on GPU, we have to expose different levels of parallelism in order to take advantage of the massively parallel computing power of the GPU. So for these reasons, we expose two different levels of parallelism. The first one is the intra-task parallelism. So we try to expose and parallelize the computation for a single pair of sequences. So we try to optimize the computation of the PRHMM on a couple of sequences, and we achieve that by mapping GPU threads to cells of the dynamic programming matrix in order to compute them in parallel, since the dependencies are defined and limited. Also, because dependencies are limited, we implemented a caching mechanism that allows us to store only the necessary data at that specific iteration of the algorithm. The other level of parallelism we exposed is the inter-task parallelism. And so we try to optimize the execution of more instances of the PRHMM in parallel. And we achieved that scheduling more instances in the kernel. And we did that by mapping a pair of sequences to a block, to a GPU block. Then we implemented a methodology that is the dynamic memory swap, that is the one that allows us to process sequences regardless of their length. And this mechanism is based on the one main observation, is that we can compute the memory required by our alignments on the host and then schedule the proper memory resource on the device. So when the host receives as input an alignment, we can compute these requirements and we can evaluate them against a threshold that is device dependent and can change with different execution if we change the device. So once we compare it against the threshold, then if these requirements are below the threshold, then we are going to use for that specific alignment on that specific block on the GPU, we are going to use shared memory, which is a scarce resource, but is requires a very low amount of clock cycle to access data. On the other hand, if the requirements are above the threshold, then we are going to use global memory, which is slightly slower in access, but is a much more abundant resource. So overall, this mechanism allows us to adapt the size and the type of memory depending on the input that we are going to process. And this allows us to overcome the limitation in the state of the art regarding the length of the sequences. And the only limitation that theoretically we have is the amount of global memory on the GPU. Finally, we integrated our solution into haplotype caller, and we achieved that by leveraging the Intel Genomics kernel library. and we plugged our solution into haplotype caller. And in order to minimize the impact of data transfers between the host CPU and the device, we provided our system with multiple computing streams that allow to hide the data transfers with the kernel computation. So this allows to optimize the execution time and reduce the overall time required for the computation. Concerning the experimental evaluation, we tested our design on NVIDIA V100 and A100, and we tested the software on an Intel Xeon Platinum 8167. In this talk, I will present you two evaluations. The first one is the evaluation of the dynamic memory swap, and then a comprehensive evaluation of the integration process. So firstly, focusing on the dynamic memory swap, here I report two plots for the two different GPUs. And in violet, it's the throughput achieved by our solution using the dynamic memory swap, whereas in yellow, there is the throughput achieved by only leveraging global memory. The red line represents the limit in the state of the art in terms of sequence length. So we can see that for short sequences, we can achieve a pretty high throughput. And this throughput remain high as we increase the length of the sequences, whereas for the dynamic memory swap, whereas for the global use only, for the global memory use only, the throughput continues to drop. But most importantly, we can achieve a very high throughput, overcoming the limitation in the state of the art and processing, still using shared memory for longer reads. And at the end, the throughput drops and follows the same trend as the global only, because in this situation, all the sequences require memory that is above the threshold, and therefore we are forced to use global memory. But this scenario is rather unlikely, because in which we will most likely have an hybrid situation where some sequences are going to use global memory and other shared memory. So we are able to mask the computation of sequences using global memory. Then moving to the evaluation of the integration into haplotype caller. In this table, I report the execution time for the tool, for the standard version of the tool provided by GATK, that is the first line in the table. Then a multi-threaded evaluation using GKL, using their optimized implementation. And finally, the execution time for our solution targeting the V100 and the A100. As we can see, moving from the original version to the optimized version provided by GKL still provides some improvements. And the execution time lowers as the number of threads increases, despite a drop using 104 threads. But then moving to a GPU accelerated solution further pushes performance, and we are able to achieve 1.3x over the single-threaded version of GKL using the V100, and more than 2x using the A100. It is important to notice that in this evaluation, we considered real data, not synthetic data as the previous one. And we used real data from a human chromosome and a whole genome sequencing sample. So this represents a very a real life scenario that we might incur to. So this proves the effectiveness of our approach by achieving speedups up to 2x. So to conclude, in this talk, I presented a high-performance GPU-based accelerator for the PRHMEM with two different methodologies, one for optimizing memory usage and enabling processing of sequences regardless of their length, and on the other hand, another methodology to optimize data transfers and achieve a seamless integration into haplotype caller. Future works will focus on further optimizations in the integration, and also I would like to try to apply the same methodology to other tools, other variant callers available in the state of the art. So that's all on my side. Thanks for your attention. And if you have any question, I'm more than happy to answer.
Thank you, Beatrice. So can you elaborate a bit more on the comparison with the state of the art?
Yes. In this today, I only presented the comparison against the original tool and GKL, but there is a variety of implementation in the state of the art that target hardware accelerator. And our implementation is able to outperform by 2 times the best performing FPGA-based acceleration, and by almost 10x the best GPU-based solution. So this proves also the fact that the dynamic memory swap is very effective also in improving the performance of the PeriHMM.
Thank you so much. So after this talk, we have concluded this first part of our event. So I would like to thank once again all the speakers for IT for Life Science. And if in the audience do you have some question, just write it in the chat and we can make contact with the speaker and let you have the answer. And if you have interest in some of the talk that you have heard or in the work that we have presented, just contact us and we can find a way to collaborate and see where our research can go with your help. So thanks again, and we will come back with the next session that is domain-specific computing in around 25 minutes. Thanks.